Now next question, that is question number 10. In this question, not true. First, prokaryotic cellular organization, this is true. Prokaryotic cellular organization, the only kingdom which includes prokaryote, this is true. Ribosome 70S type, this is also true. Presence of membrane bound cell organelles, no. Presence of membrane bound cell organelle. In any prokaryotic cell, they don't have membrane bound cell organelle. So third one is not true. Third one is wrong. Whereas cell division is by mitosis is right. Amitosis is right. So this is the correct answer. That is third one. That is, they don't have membrane bond cell organelle. And here what is written? Presence of membrane bond cell organelle. Membrane bond cell organelles are found in eukaryotes, and eukaryotes are kept under 4 kingdoms. That is Protista, Plantae, Animalia, Fungi. All 4 kingdoms, they include eukaryotes, and in eukaryotic cell, membrane bond cell organelles are found. In prokaryotes, membrane bond cell organelles are not found. That's why the third one is wrong. Then most primitive prokaryote, that is archaebacteria, responsible for the production of biogas, that is methane from the dung. They are found in the ruminant animals, and in this series only methanogens, that is methane producing archaebacteria. Methanogens means methane producing archaebacteria. Methane producing archaebacteria. So answer is methanogens. Here biogas is for the methane. Biogas is for methane. And in 12th class you have already studied, that is biogas plant is there, and then biogas plant dung is used, cattle dung is used for the production of biogas, and in that dung archaebacteria are found and they produce methane, approximate 50 to 70%. The gas emitted from the dung contains methane, methane gas, that is biogas, gas of biological origin. That's why biogas. And primitive prokaryote, that is archaebacteria, and there are 3 types of archaebacteria, that is methanogens, thermoacidophile and halophiles, and out of them methanogens are responsible for the production of biogas methane. 12th question. Wall-less, that is cell wall is not found but smallest living cell. Cell wall is not found but it is the smallest, that is mycoplasma. Mycoplasma, number 1, smallest cell of living world. Smallest cell of this living world. It is smallest prokaryote also. Smallest prokaryote without cell wall. Smallest prokaryote without cell wall. And can survive in oxygen free environment also. Can survive in oxygen free environment also. So wall-less prokaryote, here algae is with wall, bacteriophage is non-cellular structure. Bacteriophage is non-cellular, so there is no possibility to consider bacteriophage as cell. Bacteriophages are non-cellular. Cyanobacteria prokaryote with cell wall. Cyanobacteria prokaryote with cell wall, whereas algae eukaryote with cell wall. Eukaryote with cell wall. So answer is mycoplasma. Mycoplasma is also called PPLO. PPLO, pleuropneumonia-like organism. PPLO, that is pleuropneumonia-like organism. Then, when a spiral bacterium has only one curved and coma-like, let's say, better, that is bacterium, not a spiral, has one curved coma-like structure. First one is a spiral shape, second is coma-like, third one is rod-like, bacillus is rod-like, and spirochete is also spiral. So, correct answer is this one. One curve or coma-like structure, that is Vibrio. Vibrio, one very good example is Vibrio cholerae. Vibrio cholerae, that is cholera causing bacteria. Vibrio cholerae, cholera causing bacteria. Bacillus is rod shaped bacteria and is a most common shape of bacteria. Most common shape of bacterial cell, it is bacillus. Then, in addition to nostoc, nostoc is a blue-green algae, heterocysts are seen in. When we see the nostoc filament, cells are bluish green. Nostoc cells are bluish green cells like this. This is nostoc filament. Then in this particular filament, you'll see a large cell which is non-green. This is heterocyst. Whereas other cells are bluish green. And you'll see the nostoc filament, there is a mucilaginous covering over it. Mucilaginous covering is there. Most of the cells are bluish green except this one. This non-green cell and comparatively larger cell is known as heterocyst. And this heterocyst nitrogenase enzyme is present. In this heterocyst, nitrogenase enzymes are found. That's why blue-green algae are able to trap atmospheric nitrogen and convert it into nitrogenous compound. Because of presence of heterocyst, blue-green algae are able to fix atmospheric nitrogen into ammonia into nitrate by this particular enzyme, and it is contained in the heterocyst only. So, here we see Azotobacter is a bacterium, not a blue-green algae, and it is a single-celled. Spirulina, it is a filamentous bacteria. Then Anabaena, that is Anabaena. Anabaena is very similar to nostoc. It has heterocyst. So, the answer is, along with nostoc, Anabaena, both have heterocyst in their filament, and that is responsible for the reduction of atmospheric nitrogen. So, these two cyanobacteria, that is nostoc and Anabaena, they have two major roles. That is, they are photoautotrop, they are photosynthetic autotrop. Anabaena and nostoc are photosynthetic autotrop. As well as diazotrop, that is able to fix atmospheric nitrogen. Diazotrops is a word for those, that is, who are able to fix atmospheric nitrogen. Those who are able to fix atmospheric nitrogen, they are called diazotropes. So nostoc as well as Anabaena both are number one photosynthetic autotropes, and number two they are diazotropes also. They are diazotrope. Diazotrophy means biological nitrogen fixation. Diazotrophy. A process. Diazotropy is a process. What is that? Biological nitrogen fixation. Biological N2 fixation is called diazotrophy. Biological nitrogen fixation. So, specific cells in their filament, large non-green cell. These cells are large and non-green, heterocyst, and one more speciality is with them, they have only pigment system 1, no pigment system 2 with them. So they are not able to perform Calvin cycle also. They perform incomplete light phase because of presence of PS1 only. PS2 is not there, number 1. Second, not able to perform atmospheric carbon dioxide reduction by photosynthesis. Other cells can do. That is, other bluish green cells in nostoc as well as Anabaena are capable of performing complete photosynthesis except heterocyst. Again, heterocyst, larger non-green cell, larger and non-green cell, and it has nitrogenase enzyme, so able to fix atmospheric nitrogen, but not able to perform photosynthesis. So answer is Anabaena. 15th, nostoc and Anabaena I have already mentioned in previous question. I have given the complete description. That is, they are photoautotrope as well as diazotrope. Photosynthetic autotrope, that is normal cyanobacteria. Cyanobacterial photosynthesis is higher plant-like photosynthesis. That is, cyanobacteria perform photosynthesis which is higher plant-like. Higher plant-like photosynthesis. Though they are prokaryote. Prokaryote. Higher plant-like photosynthesis. Then what is diazotrophy? They convert atmospheric nitrogen into ammonia, that is nitrogenous compound. So that's why the answer 4 is correct. Answer 4 is correct. Then this structure is, I have already described, this is nostoc filament. This is nostoc filament. See all the cells except this one. This is heterocyst. This non-green cell is heterocyst. Other cells are bluish green. One speciality, you are seeing that filament looks like beads in a string. That is moniliform filament. Nostoc filament is moniliform filament, that is looks like beads in a string. Moniliform filament, that is beads in string. Just there it is just like that appearance. So it is moniliform filament. And cells are prokaryotic. So that structure which is in the picture is nostoc. It is nostoc. So the answer is nostoc. Consider, these are 4 points are given and we have to choose the correct for mycoplasma. This question says in above these points we have to select the correct options. First, PPLO, right. So this is 1 is correct. 1 is correct. Then second, cell wall is made up of. Cell wall is not found in mycoplasma. Mycoplasma is without cell wall. Mycoplasma is without cell wall. So second wrong. Third, chemolithotrop. No. They are not able to prepare their food material. They are not able to prepare their food material. They are heterotroph only. They are heterotroph only. They are heterotroph only. Causative agent of little leaf of brinjal, this is right. So first and 4 right. 2 correct options, first and 4. So answer is 1 and 4. 2, number 1, cell wall is not found in mycoplasma. Second, they are heterotroph, not chemolithotrop. Chemolithotrop, that is chemosynthetic autotroph. But they are not autotroph, they are heterotroph. That's why only 1 and 4 are correct. Then, the non-photosynthetic protestant, kingdom protesta, that is kingdom of unicellular eukaryote. It is kept under 3 categories. Kingdom protesta is divided under 3 categories. First one, photosynthetic protestant. First one is photosynthetic protestant. Photosynthetic protestants. In that case, diatoms, dinoflagellates and euglenoids are included. Diatoms, 2 dinoflagellates, and third euglenoid. Second is fungal protestants. Second is fungal protestants. They are heterotroph and only slime molds are included. Fungal protestants include slime molds only. And third category is animal protestants. Animal protestants, they include protozoa only. Protozoa. So let's see. In the question says non-photosynthetic protestants. Non-photosynthetic protestants, in that case, first option, dinoflagellate are photosynthetic, diatoms are photosynthetic, only slime molds are non-photosynthetic. That's why this is not the answer. Second, again, diatoms photosynthetic and these 2 are non-photosynthetic. That is, again, this is not answer because diatom is included. Similarly, in third one, dinoflagellate, diatoms and euglenoids, these are photosynthetic protestants. So this is also not the answer. The correct answer of non-photosynthetic protestants is slime molds and protozoa. Slime molds and protozoa. Choose incorrect. Diatoms, that is golden algae, chrysophyte, right. Gynolox, it is a dinoflagellate, right. Plasmodium is protestus. Plasmodium kept under slime mold, this is also right. That Plasmodium is different from the Plasmodium of protozoa. Actually, Plasmodium is diploid unicelled structure, diploid unicellular structure and multinucleate. Actually, slime molds are of 2 categories, unicellular, uninucleate, haploid, that is 1, then multinucleate unicelled eukaryote, that is plasmodial slime mold. Puccinia is fungi. Puccinia is fungi, but it is not Protestant fungi, it is true fungi. So here the correct answer is Puccinia. Puccinia is correct answer here because Puccinia is not Protestant. It is the member of true fungi. It is true fungi, not Protestant fungi. It is true fungi. Puccinia is true fungi. Then Kisilghar or diatomite or diatomaceous earth. Diatoms they have silica cover over the cell. They have silica cover over the cell. And they are just like a soap box like orientation. Soap box like orientation is seen of these covers. This is epitheca. This is hypotheca. And both are silicaceous covering. Both are silicaceous covering. And within this structure, the true cell is present. That is, cell wall is impregnated with this silica. That red colored structure is silica. Silica cover over the cell. Silica cover over the cell is feature of diatoms. When diatoms die, silica is indestructible. Silica is indestructible. It is deposited at the base and constitute diatomite. It constitutes diatomite. So this is correct. Deposition of dead diatom cells, right. It is used in the making of soundproof room. It is also right. It is used in oil refineries. It is also right. So all three features are correct. That's why answer all the above. That is, what is the role of, what is the use of diatomaceous earth or Kisilghar? In this respect, all are correct. All are correct. Then red tide. Red tide, you must know. First one, Red Sea. Red Sea. Red Sea is caused by Trichodesmium. Trichodesmium erythraeum. And it is Monera. It is a member of Monera. Then red tide. Here the question is asked. It is caused by red dinoflagellate. Red dinoflagellate. Red dinoflagellates are responsible for the causative agent of red tide. That is, they are of different color. Actually, dinoflagellates are of different colors. Some of red dinoflagellate, they come with the tide. They come with the tide. The oceanic area, they come with the tide. So they constitute red tide. So again, the answer is first one. Answer is first one. That is, red dinoflagellates are responsible for the red tide. And third one, more information I would like to share. Sorry. Third information, that is red snow. Red snow. So three terms in which red word is common. Red sea, red snow, red tide. Red snow is caused by Chlamydomonas species. Chlamydomonas. Nivalis. It is found in the snow area and impart red color to the snow. Chlamydomonas nivalis is found in the snow. Cryophytic algae. Cryo, that is extreme cold. and impart red color to the snow, hence responsible for red snow. Then on the basis of nutrition, euglena. Euglena is a unique feature. It has unique feature in what respect? That is, first of all, euglena, unicellular eukaryote. It is a spindle shape structure, spindle shape with a small opening towards upper area, due to which it responsible for the ingestion of various organisms through which it can ingest. Then one more speciality with the euglena, that is, they have chloroplast also. Euglena have chloroplast also. In euglena, chloroplast is also present. So because of presence of chloroplast, ability to perform photosynthesis, but it can also perform animal like nutrition, that is ingestion through mouth, ingestion through this cytostome, ability to ingest material through this structure, that is known as cytostome. So dual mode of nutrition, dual mode of nutrition, dual mode of nutrition. Euglena is known for dual mode of nutrition, that is animal cell like as well as plant like, animal as well as plant like. Hence euglena is mixotrop. Euglena is mixotrop. Euglena is mixotrop. Now, in which of the following the cell wall forms two thin overlapping cells which fit together as soap box? Just in previous two questions, I have already mentioned in in diatoms, the outermost covering is silicaceous, and the silica is oriented in this manner, that is just like two overlapping shells made up of silica. Upper one is epitheca, and the lower one is hypotheca. This is the silicaceous covering in soap box like manner. Silicaceous covering. Silicaceous covering and indestructible also. The outer one is epitheca. First one is epitheca, and this one lower one is hypotheca. This is hypotheca. Found in diatoms. This is cell, and red one indicates the silicaceous covering. The soap box like orientation of silica cover is seen in diatoms. Clear? Upper cover epitheca, lower cover hypotheca, and it is indestructible. That's why after the death of diatoms, it accumulates at the base of water body and constitute diatomaceous earth. So answer is chrysophytes. It includes diatoms and desmate. Chrysophyte is the correct answer. In dinoflagellates, cellulose is present in the form of cell plates. In dinoflagellate, cellulosic plates are there. Cellulosic plates over cell wall. Euglenoids, they have outer covering pellicle. that is true cell wall is not present, proteinous covering is there on euglenoid. In slime molds, no cell wall in vegetative structure, no cell wall in vegetative structure.